Okay, thank you, James, for the introduction. It's my pleasure to discuss my PhD research with you guys. So I basically will be discussing exosome polymer hybrids, which is a major chunk of my PhD thesis at Carnegie Mellon. I work with Professor Mereshevsky, who is a giant in polymer science, and then Professor Das, who is a magician with nucleic acids. And I somehow was able to combine these two skill set with exosomes. So going further, okay. There's a slide issue. Okay, so we all know about exosomes. These are these tiny particles which are secreted by all cell types in the body. The size range is essentially 40 to 160 nanometer, and they're found in all sort of body fluids. Blood, saliva, tears, cerebrospinal fluid, and many others. And since they're being secreted by cells, they carry the cells specific bioactive cargo. And the key properties, which is important to me in a way, is that they can easily internalize inside cells, and they have this ability to cross the blood-brain barrier, making them excellent candidate for drug delivery. They have several mechanisms. They can either they can undergo this direct fusion, or they have multiple mechanisms to internalize, or in fact they can have this surface interactions to induce some sort of mechanisms inside the cell. So it's been 30 years. So they were first discovered in 1980s, but up then people used to believe that they are garbage bins, and cell is essentially dumping anything it doesn't need. But it took us 20 years to realize that they're involved in both health and disease, and now they are prime candidate. And there's active research going on in the field of diagnostics and drug delivery using exosomes. But the field is very new. There are lot of mysteries, up from from how they're secreted, how they're sorted, why the size is so heterogeneous, to the point how does it interact with the recipient cell, and what are the real functions in health and disease. And these are, I would say, are the mysteries for the biologist. I'm more concerned in terms of the application for drug delivery, and there are certain challenges which need to be addressed. One is there are limited tools for functionalization. There exists no versatile method where you can easily functionalize them. Stability is a big issue. They tend to aggregate with time. At 4 degrees Celsius, they'll keep clumping. At 37 degrees Celsius, they tend to shed their surface proteins. So the integrity is compromised. And more importantly, when you after the systemic administration in the blood, they are washed out in 3 hours. There are reports which say even 2 minutes. So there is a strong need to build a robust system, and our idea was to combine them with polymers, because polymers are really robust systems. But how do you really bridge this world of polymer science and exosomes? And in fact, James published a very beautiful review recently in 2017, where he discussed all the mechanisms, all the strategies which are being used. So mostly they rely on the cell source. So either you can go for the genetic manipulation to load some sort of mRNA or some non-native proteins, either inside or on the surface. Or the alternative strategy could be metabolic labeling, where you feed the cell with some artificial amino acids or sugars and end up expressing some chemical functionalities for further reactions. And then these other areas as well, but none of these really fit our need to basically bridge these two systems other than the hydrophobic insertion. Hydrophobic insertion, it's a non-invasive technique where you, by functionalizing the system with a hydrophobic unit, you can anchor it into the membrane. Exosome membranes are very different from cells. They're not as dynamic. Also, they have high amount of cholesterol in there, which essentially, make them more rigid. So developing on this, we developed a method which was independent of the cell source. So what you do is you essentially functionalize the one end of the DNA with a cholesterol moiety, add a spacer in between, and the other end of the DNA can be functionalized with any chemical functionality. It can be a dye for bioimaging, or it can be some small molecule for other interactions. The process is simple. You basically do a gentle vortex with these two systems for 5 minutes, and now you have decorated exosomes with these chemical functionalities on the side. But the other advantage of using DNA, the question comes, why DNA? Because the negatively charged phosphate backbone really allows you to orient them outside, because there will be static repulsion between negatively charged membrane and the DNA. So most of them will be exposed outside. And then, you can further expand on this advantage of DNA. You can go for complementary sequences and essentially bring in another functionality on the surface. And through that, we were able to attach, dyes, aptamer, antibody, and other small molecules, which, have also been explored from through other strategies. And we wanted to really characterize them thoroughly. And we saw, depending on the concentration, you can put almost 2,000 to 7,000 DNA per particle. We checked for a week. This linkage was stable under storage, both temperature. And so basically the cholesterol doesn't fall off. And most important was, that whether we were able to preserve the cell uptake. And, that worked really well for both single standard systems and double standard system. Next, so we published this paper, we further explored them, by printing and basically showed that they're active on the solid phase as well. But, moving further with this goal to prepare a robust exosome polymer hybrid. Now we can easily guess that if we have access to a DNA polymer hybrid, basically it's a block copolymer. If you have access to this, then you can easily functionalize these systems both by annealing approach and pre-annealing approach by simple vortexing for 5 minutes. And we did that. And we, in fact, looked into different monomers. So this is a peg-like system. So it's not a peg, it's a peg brush, where each side chain of the polymer chain is a long peg molecule. So this is very different than which we commercially see in other many products. And we also explored zwitterionic systems, which are highly biocompatible, non-fouling. And then we had this another variant in our lab, which is DMSO derived. It's a special monomer because it's being considered as an alternative to PEG. It's highly water soluble, and now we are seeing reports where this monomer itself has some intrinsic bioactivity of targeting to macrophages. So we characterized them very thoroughly. So we had collaborators in Germany, and we looked at through field flow fractionation, and we looked at the rho parameter, which essentially tell you whether the particles are spherical or not. And what you see is that with increasing polymer loading, the rho parameter decreases, and it's more depictive of spherical vesicles, which have long antennas on the surface. We checked, depending on the our quantification study suggested that we can go as high as one chain every 8 nanometer square area. And depending on the concentration, you can have from 2,000 to 8,000 polymer chains per particle. So this is a very high loading. And it comes because, our applications are in biology for drug delivery. Optimizing this loading is very important because we know that the surface proteins on the surface of exosomes are very important for the interactions. And we wanted to make sure that those are accessible. So for that, we dive deeper, and we use anti-CD, it's a flow cytometry assay. So we use anti-CD63 beads. CD63 is a surface marker. It's present on exosome surface. And we were trying to look at the binding on the beads after polymer functionalization. So we looked at 3 candidates, 3 different lengths of polymers, 10,000, 20,000, 30,000. And by playing with the ratio, because it's this method allows us a multivalent functionalization. By playing with the ratio of these 2 systems, we were able to control the loading, but kept the dye concentration constant on each particle. And now, easy way to visualize would be that essentially we are increasing the loading and also, increasing the polymer length, and we're trying to see how the binding is affected by looking at the dye signal onto these beads. And we tested 3 different polymers. For smaller polymer chains, the chains are so small that the binding is not affected at all. That means the surface is still accessible and we see no compromise. But if you go to longer polymer lengths, in that case, the binding sort of decreases with increasing loading. And for 30,000, there's a good compromise. And then, if you go to higher loading, it keep on further decreasing. But another key aspect over here is if you look at these systems, we have DNA sitting there. And, there are nucleases in the body and they can degrade the DNA. So the stability of the DNA is also very important. So for that particular reason, we basically treated these beads, exosome-bound beads with DNAs. And in that case, if the DNA is accessible, we should be able to see a drop in the dye signal. And that's what we saw for smaller polymers, low loading, there's just essentially no polymer to evade the nucleases. And in that particular case, almost 80% of the DNA and polymer is cleaved. But if you keep on increasing the loading, you start regaining the stability. For longer polymers, beyond a certain loading of 1 micromolar, you start seeing complete stability. That means even if you treat it with nuclease, the polymer does not really allow the access. And for longer polymers, lowest loading, complete stability. And so stability of the DNA was very important to us. So we stick to 30,000 for all our future studies, and then dive deeper into bioactivity assessment. We wanted to make sure that they are still, the cell uptake is conserved. So we looked for different polymer lengths, different polymer types. We also tested it with inhibitors, so which can inhibit the major pathways for exosome internalization. And in that case, we were able to see similar drop, confirming that we are not really tweaking the internalization pathway as such by polymer. And that was in a way our goal. We also looked at some native and non drug loaded exosomes. So we use mesenchymal stem cell derived exosomes and then functionalized with polymers, and then looked for the enzyogenic property. Then also we loaded the exosomes with BMP2 growth factor, and then looked for the osteogenic differentiation using these systems. And finally, we loaded them with curcumin. And in all the cases, we were able to see no difference in the activity of exosomes and the exosome polymer hybrids, sort of really confirming that we are not compromising the bioactivity. Then we looked for the physiochemical properties. So first, the idea was to enhance the storage stability. So we incubated native exosomes and polymer functionalized exosomes for 30 days, and we were able to see very conserved size profile for exosome polymer hybrid. However, native exosomes tend to aggregate. We saw that within size increase, and also we observed protein shedding at 37, and some sort of clumping as well. But then, what do you do after 30 days? What if you really need to remove the polymer at some point? So in that case, DNA, we can, as I said, magician of nucleic acid, we can easily put a photoclable group in between, which will really allow you to remove the DNA and hence the polymer from the surface by just irradiating light, UV light for 2 minutes. And we sort of tested this beautifully with another experiment where we assess the stability of exosomal surface proteins against trypsin. So if what we saw was that this is a size exclusion profile. We radio labeled the surface proteins of exosomes. So if they're stable or in the absence of trypsin, all the radioactivity signal will be in the exosome fraction. And that's what we saw both for our native exosomes without trypsin and our exosome polymer hybrids. But if you shine light for 2 minutes, in that case, the polymer is cleaved, and now the surface is accessible to trypsin, and you see this nice shift up with the radioactivity into the later fractions. Next, after testing this ex vivo, we basically moved in vivo, and we looked for blood circulation, and our exosome polymer hybrids, in fact, had a very superior blood circulation. Where the native species were washed out or basically out of the bloodstream in 3 hours, we had almost 30% circulation with zwitterionic polymers in the bloodstream. Another important key thing was tissue distribution, because polymers can really tweak the targeting properties of their system. So we looked at the percentage accumulation after 24 hours in mice. And what we saw was there's, we in fact had a similar percentage accumulation. Although they're still in circulation, the absolute values are very different, but the percentage was still similar. So we preserved the tissue targeting properties. So just to, switch gears, till now I essentially discussed this strategy, grafting 2, where we need access to this DNA blocker polymer. But another powerful strategy can be grafting from, which is more challenging. But the idea is that you attach an initiating moiety onto the surface of exosome and then starts growing polymer chains right from the surface. And this is doable because another chunk of my PhD thesis is basically making DNA initiator, where you basically attach a carbon-bromine bond, which act as an initiator for the polymer synthesis by atom transfer radical polymerization. So we combine this, and in fact, we were able to achieve it. But it's not as simple because the polymer conditions need to be really biocompatible. Now, what you're doing is these are not natural polymers; these are synthetic systems. So, and exposing exosomes to synthetic systems which has copper and other sort of diluents, in that case, it can be really challenging. So we use glucose oxidase mediated oxygen tolerant ATRP. In that case, you don't expose exosomes to any rigorous degassing procedures, and we stick to blue light to sort of have a more biocompatible method, and we essentially functionalize them with polymers. But as I said, it's powerful technique because now this allows you to even chain extend them with a different monomer. And this is a very, now you have expanded the functional profile a lot, because now you have two different polymer systems sitting on the surface. And we tested that, we were able to easily extend with one peg brush like block, and then you further extend it with another OEMA. And this also allows you to, in fact, test different monomers. So this is DMEMA, it's a cationic polymer. And let me tell you, it's near impossible to put a cationic polymer on exosomes by grafting too. So, we expanded the monomer scope through this. But the real idea behind this grafting form was to basically make biomaterials. So now, this is, let me walk through this. So basically we combine grafting from strategy with the in situ gelation. Essentially, when you're growing the chains from the exosomes, you also start making a gel with another initiator. So there'll be some 5% of the chains will be polymer chains will be growing from the hair, and they will become part of the gel network because they'll start cross-linking and become part of the gel network. So what you've essentially done is that using cholesterol-based systems, you have tethered your exosomes into the gel network. And now if you have a photoclavable system, in that case, it really allows you a photoresponsive gels. That means on demand, you can cleave off this DNA tether using UV light and essentially release all the exosomes from this hydrogels. So, to really see how this strategy or the tethering strategy will be better than physically entrapped systems. So we tested. So this is a simple growth factor protein, BMP2, and these are physically entrapped exosomes. That means they do not have any initiator. When you're forming the gel, you just simply add them into the pre-gel solution. In that case, they stay up to 12 days inside. But if you look at basically tethered exosomes, in that case, what you see is a much, much superior release profile. Even after 30 days, you had almost 30 to 40% still retained inside the gel. And if you are using the photo responsive systems, in that case, so on day 25, we irradiated them for 2 minutes with UV light, essentially breaking all the tethers and releasing all the bound exosomes. So just for the proof of principle, essentially what we did, we wanted to look for osteogenic differentiation using BMP2. So we tested two systems. So this is an early marker for bone differentiation. And this side essentially tells you solution phase. That means you're adding all the or your stocks to the cells. And over here, you're essentially keeping a gel piece with the cell. In that case, we saw, early differentiation, but the main key thing was that, if you start moving to late differentiation and look for calcium deposits, in that case, only in the case of BMP2 loaded gels, we were able to really, reach the late differentiation marker. And if you compare these two systems where you need to add fresh stocks every 72 hours versus just keeping a small piece of gels, this was like a way superior method. So finally, to summarize my talk, we prepared exosome polymer hybrids using an on-demand system, using cholesterol-based DNA tethers. We have a very precise control over loading, and we are able to preserve the intrinsic bioactivity. We enhance the ex vivo and in vivo stability, and and moving further, one thing would be, can we take the systems across the blood-brain barrier? And for the second part, we prepared these exosome hydrogels, which are more, using synthetic polymers. So they're highly tunable as compared to, other natural-based systems. And we have a very interesting or superior, I would say, release profile because of the tethering strategy. And, nothing is enough. Can we basically, from 2 months, can we go to 6 months release profile? And we also basically want to check them in vivo in terms of the bioactivity. So with that, I'd like to summarize my talk. My advisors, Sai, who is my partner in crime in all these exosome projects, and all our collaborators, who helped us, throughout these projects. Thank you.
Thank you, Sushil. That was fantastic. Yeah, so if there's some questions coming in, but if anybody else has any other questions, please post them on the chat. We have a question from Ryan who says, wonderful talk, Sushil. Could these exosomes be packaged up and transported from one lab to the next?
Yes, we did that. So in fact, our this field flow fractionation, they were done in Germany, and we shipped them from here after polymer functionalization, and they were intact. So yeah, they
I guess maybe as a follow-up to that, does the tethered gel approach offer an approach to transportation rather than sort of cold transport or anything like that?
I see. I mean, yeah, yes, we tested them in vitro, and I would say, I mean, if they're able to retain for such long time, yes, transportation should be convenient. And it depends, what kind of release profile you're looking at. So these gels are highly tunable, so you can control the mesh size. And let's say if you transfer them with the photocleavable tethers, they should stay inside. And once they reach there, you can essentially cleave the DNA tether and remove them, or like basically release them as a physically entrapped system.
Okay, thank you. So I have a few questions as well. So just a clarification, first of all, with the exosome tethered gel,
Yes.
with the osteogenesis you were showing, was that a 2D culture? Was that a 3D culture? So were the cells embedded or were they on top of the gel?
No, they were on top of the gel. Yeah, it was like essentially, if you could see the pictures here, they were on top of it. Yeah.
So is there any way of adapting the system for a 3D culture?
Definitely. I mean, it's just that, it's possible, but since I'm graduating, yeah, I will have to sort of, figure out how to continue this. We're also thinking in terms of like injectable systems using these. But yeah, if you have some access to 3D print, 3D cell cultures and everything, yeah, it's doable.
Well, I was just wondering in terms of the kinetics, how fast is the gelation? Maybe you showed it, but maybe I missed it.
Like you basically shine light for 60 minutes. So it's you make a pre-gel solution and irradiate light for, yeah, you blue light for 60 minutes.
Okay, sure. So it might be a bit slow unless you have a good viscosity precursor. That
Yeah.
maybe we can follow up on that. But we have a question from John who said, "What are your limitations when making hydrogels around exosomes? Is there a maximum concentration of monomer that you could use without disrupting the endosome?" Or maybe he meant exosome.
Yes. So yeah, I mean, we tried. Okay, you need to make sure that the exosomes are sitting comfortably in there. So you cannot have really tight mesh size. So our mesh size is in the range of 200 nanometer, or like 200, 300 nanometer, basically making sure that exosomes can easily sit there. I think that's the first, should be the, concern when you're making these. And second, I would say, so what was the question again? Like, in terms of limitations, right?
Is there a maximum concentration of monomer that you could use without disrupting the system?
Yes, so we basically worked at 40% concentration because when you're making gel, you use a high amount of monomer for sure. But it depends, like how long are you exposing them? So like monomers, certain monomers are cytotoxic as well. And in that case, if exposing them for too long, they might penetrate inside and turn out to be cytotoxic, or somehow compromise the integrity of system. So it depends really on the monomer, what monomer you're using, and that will basically define your concentration.
Okay, that's fine. We have a question from Rodrigo. He says, "Thank you for the talk." So I actually think we touched on this, but he asks, "Is it possible to load the exosomes with other drugs, like in liposomes? Did you try loading the exosome with other drugs, such as anti-tumoral drugs, for drug delivery in cancer treatment?"
Yes. So, I mean, it's our collaborators are essentially exploring, those aspects. Mine was like mostly on the material side. As a proof of concept, we tested this growth factor and curcumin. Curcumin is in fact in a way anti-cancer. But yeah, I mean, we need to explore further and, go for a more, goal-oriented material. Till now, we're more exploring, can we really have a well-defined, material?
And what was your mechanism for loading BMP? Actually, I was curious.
Yes, it was. So it was done essentially through collaborators, but I think it was just very simple sonication.
Okay.
Yeah.
We probably have time for maybe one more question. So this is from Joanna, who says, thank you, Sushil. Very nice talk. Have you quantified the uptake efficiency of your exosomes by cells? And then what percentage of the exosomes that you deliver are actually uptaken in intracellular?
Yes. Okay. I can go back to slide. Yeah, we tested like different systems. So yeah, if you see here, there is some compromise if you go to longer polymers, but these are done in hex cells after 6 hours, and we in fact see almost 60% internalization. So there is some compromise, and it can be somehow using different polymers you can maybe counter it at some extent, but the rate of it's after 6 hours. So I would say efficiency is slightly lowered, but it's not compromised to that extent.
So it's 60% of the cells are labeled. Is that correct?
So exosomes, yeah, I mean, yeah, exosomes
So
were labeled and we see quantified it from the cells, and we saw 60% uptake.
okay, so I think that her question, maybe it's a little bit difficult to answer actually, but in terms of the percentage of the exosomes which are uptaken, so are there free exosomes which are not uptaken by the cells?
I see. Um, yeah, I mean, that's a good question to look at. People observe recycling as well. They say that exosomes tend to recycle as well. Yeah, but we still need to look into it. That's nice question.
Yeah, it's a quite a tough thing to quantify.
Yeah.
So yeah, I think that's all we have time for. So